Good afternoon, everyone. Today, we are honored to have Dr. Jesse Jokerst to give us the lecture. Dr. Jokerst is an associate professor in nanoengineering at UCSD. So he earned his PhD in chemistry at UT Austin, and he completed a postdoc training in Stanford radiology. His lab is focusing on projects related to human health and nanotechnologies. Dr. Jokerst won many prestigious awards, including NIH Pathway to Independent Award. It's also called K99, NIH New Innovator Award, NSF Career Award. Without further ado, let's welcome Dr. Jokerst to give us a lecture on photoacoustic technologies.
Great. Thank you, Lingyan, and thank you everybody for listening. So I have actually taught this course before, but it's been a couple years. And so this is very informal, so you can, if you want to just unmute yourself, I don't know if you can unmute yourself and ask me a question or send a chat, feel free to do that. So yeah, I think Lingyan introduced me well, that my background is in chemistry, and so a fair amount of what we'll talk about today is kind of contrast agent development and applications, but I also want to talk about some of the basics of the interaction of sound and light. And so that's basically what we're going to discuss today, is how does sound and light interact with each other. So I first want to start with this slide and talk about molecular imaging versus anatomical imaging. So I trained in a radiology department for 6 years as a postdoc. And so when I was in that, my fellowship was called MIPS, the Molecular Imaging Program at Stanford. And so I've thought a lot about molecular imaging, and you might not necessarily off the bat understand the difference between anatomical imaging and molecular imaging. And so I'm showing here two examples. On the left, this is a chest X-ray, and on the right, this is a positron emission tomography experiment. And so when you look at these, you might think, well, these are pretty similar, right? They're both of the chest space. They've both got, black and white. There's some areas that are higher or lower, but these are actually created through fundamentally different processes, and they're showing information that is fundamentally different. So on the left, in this chest X-ray, we simply have a source, a sample, and a detector, right? It's kind of like a spectrophotometer, right? Source, sample, detector. And wherever absorbs more X-rays versus absorbing less X-rays produces this image, right? So the lungs are pretty low density, so they don't absorb a lot of X-rays. So that's why the lungs appear dark, because a lot of X-rays are hitting the detector. Similarly, the bone is absorbing a lot of X-rays. Okay, so what we're imaging on the left is structure, bone, tumor, tendon. We're imaging anatomy. On the right, in positron emission tomography, we're creating this image through a totally different system. So here, we've created this radio-labeled glucose analog. So this is just like glucose, except that we've put this fluorine-18. This fluorine-18 is going to emit gamma irradiation, and these high energy gamma rays are then detected with a positron emission tomography system, and we're creating this kind of image. So what we're imaging here then is, where does this fluorodeoxyglucose molecule go after it's been injected into the human subject? Okay, so what uses a lot of glucose? Well, your heart uses a lot of glucose. Your brain hopefully uses a lot of glucose. And as you might know, tumor and metastatic tumor also use a lot of glucose. So when you look at this, you might think, well, I'm imaging the brain, I'm imaging the heart, I'm imaging tumor, and that's kind of true. But what we're actually imaging in this experiment is the biodistribution of the glucose 6 transporter, right? Because that glucose 6 transporter is what is causing this radiolabeled glucose to pass across the bloodstream into the cells and accumulate in the cells. So that's why people call this molecular imaging, because what we're imaging here is molecular level information. And so molecular imaging has expanded beyond PET to a variety of imaging modalities, but by and large, it remains a PET technique, an optics technique, and in some areas, an ultrasound technique. There is molecular imaging in MRI. It just tends to be, it's more rare. MRI tends to be more of a kind of an anatomical technique. So I got interested. Oh, let me just comment then briefly that molecular imaging frequently uses some kind of contrast agent. So if you hear me say contrast or contrast media or contrast agent, it all kind of means the same thing. And they come in a bunch of different designs, but kind of at the heart, there's some kind of reporter and some kind of targeting agent that causes this entire contrast agent to go to the biology that you're interested in. And so in fluorodeoxyglucose, the entire small molecule is basically the targeting agent. And the reporter is this fluorine 18 atom. And then it has a simple covalent bond. So I'm in the nanoengineering department here at UCSD. So a lot of what we make, the reporter, maybe even the targeting agent is also a nanoparticle. But this contrast agent then links the basic biology to the practice of medicine. So I got excited about ultrasound primarily because of this figure. So when I started my postdoc, I was working on surface enhanced Raman spectroscopy, and I was had this Raman microscope, and I had these nanoparticles, and we were going to build a Raman colonoscope for doing colonoscopy, and a couple other Raman projects. And so I would collect a reference spectrum, and then I would put my sample on there. And at the end of like an hour, I could map 1 centimeter by 1 centimeter of tissue. And I couldn't go more than a few millimeters into the tissue. It was kind of frustrating. And then one day I went downstairs and turned on the ultrasound system, and it was instantaneous. I could get hundreds of frames per second. It went deep into tissue, and basically wherever I pointed, the transducer is where I created an image. And so that kind of got me excited about ultrasound, because I could overcome a lot of those issues that I had with other techniques. So what I'm plotting here is the temporal resolution. So temporal resolution means how fast can I collect an image, right? So if you've had an MRI or a CT, you're in that metal tube for quite a while, right? So that means it has a long temporal resolution, right? I'm also plotting then in vivo spatial resolution. So that's the smallest feature size that can be detected, right? So you generally, if you want good spatial resolution, you want this number to be small. And so optics scans a lot of different regions because there's so many different techniques in optics, right? Are we doing super resolution microscopy? Okay, well then we're even farther down here, right? Are you doing a reporter gene in a mouse? Well, then your spatial resolution is up here, right? So optics is quite broad. Ultrasound is also kind of broad in terms of both its temporal and spatial resolution. The spatial resolution, what do you think? I'll let you think about this. It's coming up in a few slides, but I'll let you think about what is the primary feature that governs the spatial resolution. Feel free to jump in, of ultrasound imaging. On these other ones, the temporal resolution, the spatial resolution, there's not as much control. They tend to be kind of quicker bend. The temporal resolution continues to improve, but the spatial resolution is kind of a function of the detectors. Okay, so that's what we like about ultrasound. It has good temporal and spatial resolution. It's also by far the most common piece of imaging equipment in medicine. So everywhere from, private practice clinics, rural clinics, developing world clinics, they all have ultrasound. There's really good systems you can buy now for a couple $1,000. Most people tend to think about ultrasound in terms of monitoring pregnancy, but it's also used extensively in cardiac medicine, monitoring a variety of tumors. It's used in joints. It's used in a lot of areas of medicine that the lay public might not think about. They tend to think about it just in terms of obstetrics. Okay, so that's what's good about it. What do we still want to improve about ultrasound? Well, for one, it's primarily an anatomical technique, right? And so when I showed my difference between molecular imaging and anatomical imaging, ultrasound by and large is an anatomical technique. It also has relatively low contrast, which we'll come back to, and a lack of multiplexing in terms of getting multiple color channels of information. So let's talk a little bit about sound before we go on any further. So sound is a pressure wave, unlike light, right? Light is an electromagnetic wave, and sound is a pressure wave. So since it's a pressure wave, that means it needs a medium, and it's governed very strongly by a medium, right? So you can have light in outer space, but you can't scream in outer space, right? Because there's nothing for your scream to propagate through, but there's nothing to stop you from going to outer space and shining a laser pointer, right? That would work, but you can't have a conversation in outer space. So audible range, the frequency of the sounds that your ear can hear are generally about 20 hertz. That's pretty low to about 20 kilohertz. Ultrasound, so ultra, means it's a higher frequency. So technically, people consider that things above the region that the human ear can hear, but for most imaging applications, ultrasound is around 1 to 70 megahertz. Well, we talked about this. It's a mechanical wave. It must have medium, and it's a longitudinal wave, meaning that the direction of propagation, it affects the medium in that direction, right? So as it propagates, it affects the medium and through what's known as compression and rarefaction, and we'll show that in a minute. So the speed of sound is a function of the medium that it's in, and more particularly, it's a function of the bulk modulus. So basically how compressible is your medium and the density of that medium. So something like skull bone, so the bone of the skull, speed of sound is much, much higher than something like lung. So the lung is going to have a bulk modulus that's significantly lower than that of the bone. Most tissue, people approximate the speed of sound in the human body as 1540. So that's the number that most often is used when people use ultrasound in people, is 1540. And of course, it varies in these different kinds of tissue, but by and large, that's a pretty good approximation. And then I'm just including up here some of these speeds of sounds in other media. So the intensity of sound is a function of the pressure amplitude at any point squared over the density of that medium and the speed of sound in that medium. So this is not commonly used for reasons that I don't even completely understand. The community has gotten behind thinking of sound as relative to background. And so that's when you've heard of decibels, that's what that is. So 1 bell is the base 10 logarithm of the acoustic intensity at any given point over some relatively arbitrary detection threshold, okay? So it's a base 10 log of this ratio, the acoustic intensity of interest over the acoustic intensity of the threshold detection of a human ear, okay? So what's a decibel? Well, that's just simply this, this term multiplied by 10, okay? It makes it a usable number is basically, I guess, the reason why the community has done this. Okay, so what is this sound and air typical threshold of perception of an average human? What is this I naught? The first few times I gave this lecture, I just glossed over it, and then I got interested and learned a little bit more. Well, it stands to reason that as people age, so here this is men in their 20s, men in their 30s, men in their 40s, up through women in their 60s. So different ages and genders of people. And so what you can kind of appreciate is that as people age, their hearing gets worse. That stands to reason. And by and large, people are more sensitive to higher frequencies. Okay, so higher frequencies, and there's slightly more sensitivity than lower frequencies. The exact value that the community has kind of come around, an auditory threshold at 1 kilohertz of 2 to the -5 pascals. So that's your I naught in this term, okay? And then here's just some of the decibels of conventional things that you might experience in your normal life, from a normal conversation up to a jet engine. Okay, so what does this have to do with biophotonics? Well, there's kind of two areas of interface between sound and optics. That's probably an oversimplicity, but there's two I'm going to talk today about. Briefly, acousto-optics, and then in more detail, photoacoustic imaging. So in terms of acousto-optics, what you're essentially doing is you're modulating the tissue properties using ultrasound. And by modulating the tissue properties, you're modulating the light that goes through this, okay? So one key issue is that you have this 90-degree orientation, okay? And we'll talk about why you need that in a minute. So the pressure wave, which we talked about, the sound wave, requires a medium, and it affects its medium, okay? Hopefully that makes sense. But what's pretty neat is that it's not, so what is it affecting of that medium? Or if we're in tissue, what is it affecting of that tissue? Well, believe it or not, you're actually changing the absorption coefficient, the scattering coefficient, and the refractive index, okay? So that's kind of cool. You're probably not changing it a lot, but you are, as you get these areas of compression and rarefaction, as this sound wave propagates through tissues. Is just showing the same thing in a different way. Compression, rarefaction of these peaks and troughs, you're causing subtle changes in the optical properties of tissue, okay? So in areas of compression, you're going to have more scattering, more absorption, and in the rarefactions, you're going to have less absorption and less scattering, okay? The period of this area in the tissue is going to be long, right, relative to light, okay? Because these areas of compression and rarefaction are going to be on the order of sound's wavelength, which is usually millimeters as a function of the central frequency of your transducer. So normally, if you're pushing light through the sample, there's going to be minimal impact on light because the wavelength of sound is so much longer than that of the light. right? And so that's why to get this to work, you almost have to have it at a 90-degree orientation. Okay, so this is just kind of a cartoon showing that if you have this sound, you have light, and orthogonal to light, you have sound. And so say you are tuning the absorption coefficient up or down such that all of your green and blue photons here happen to be absorbed, but the red photons are not absorbed, then the red photons can pass. Okay, so here the sound would be propagating in this direction. And so you're creating areas of high and low absorption that then in turn can cause differences in the light propagation when the light propagation is orthogonal to that direction of the sound. So obviously it's a function of the medium and the frequency of the sound. So this is just in a little more detail what this would look like. You're scattering media, the ultrasound transducer, orthogonal to that, your laser. And then this is shifted relative to the frequency of the ultrasound. So you want to have not only the physical orientation of these, but also the pulse duration to be offset a little bit from the ultrasound driver. And then just detect it with the CCD. So, what do you think trying to do this in tissue, if we're not just doing photonics, if we're doing biophotonics? Well, what is the big problem? Photons scatter, right? And so the amount of change on the photons that you will have by the compression and rarefaction is going to be much, much smaller relative to the absorption and scatter that the tissue itself is going to have, right? So tissue has a pretty high absorption coefficient and a pretty high scattering coefficient, and it varies with the tissue type, how much blood is in the tissue, how much fat is in the tissue. So to try to modulate that in a meaningful way using a sound wave is pretty difficult, okay? Because there's already so much absorption and scatter that the impact of sound I'm showing here is quite small. So maybe you can use like a very small piece of tissue to try to minimize the amount of scatter, but it has had limited impact in vivo. One kind of a success in acousto-optics is some pieces of hardware. So if you could imagine that you have a piece of a medium like quartz and a piezoelectric transducer, we'll come and talk about this a little bit more if you're not familiar with this. And by changing the central frequency of this transducer, you can change the refractive index of this quartz, and therefore you can change the angle at which this incident light emits this chamber. Okay, so that's kind of cool. But by and large, acousto-optics have had limited impact, at least in terms of in vivo biophotonics. Okay, so another kind of interaction of sound and optics is called optoacoustics, or I'll call it photoacoustics. Optoacoustics tends to be the more European term. Photoacoustics tends to be the more Western and Asian term. So here we have an incident light pulse that goes into the tissue, and we have ultrasound come out on the other side. It's magic. So let's first of all think about just creating a regular ultrasound image. We already talked about the acoustic intensity. Okay, so the acoustic intensity is incident on our tissue. Let's go here. Okay, so let's say this is our sample. this is our transducer, and it's emitting and receiving pressure waves, okay? So a transducer is simply a little thing that vibrates. This is called a piezoelectric transducer, PZT. And these PZTs are really neat materials that when you put a potential across it, they vibrate, okay? So you can cause it to emit sound waves. Similarly, if you have no potential across it and the PZT receives a sound wave, it will in turn vibrate with the same frequency of that incident sound wave and cause an electrical signal to be produced, okay? So this same material can both emit and receive a sound wave. And these transducers comes in all different shapes and sizes. I'm showing some of them here. Okay, so when my transducer emits and then I interact with the sample, this pressure wave is refracted and fracted back. What governs that? Well, it's a function of what's known as impedance, and impedance is characteristic of tissue type. And the intensity reflection coefficient describes the interface between the reflected sound and the incident sound. And it's a function of the two tissue types that the sound is traveling through, okay? So if we have a high intensity reflection coefficient, a lot of this sound is reflected back, okay? That is going to lead to more contrast in our image, okay? So let's look at something that should produce a lot of contrast. Something like skull, which is 8 to the 6th, and water, 1.4 to the 6th. So that's relatively high. But if I want to try to see the difference between blood and kidney, that's going to be pretty difficult because there's very little difference in their acoustic impedance. So this is described as acoustic impedance mismatch. So lo and behold, when I pull up an image of a fetus from Google, you can see this area of the skull having a lot of contrast with the background water, the background brain. But it's pretty difficult to discriminate between, say, the liver and the kidney because it's all the same mushy mess, right? But there's a lot of refraction and refraction differences between skull and brain and skull and water, okay? So actually, I want to briefly talk about what I mean by contrast because it can have different meanings in different contexts, and I think it's important to describe it in the terms of imaging. So let's say I have this image, I draw a line profile across it, okay? And then I plot the pixel intensity of this line profile here, okay? So the mean here is the mean of this. The mean here is the mean of this, okay? So if this is my target and this is my background, right? There's signal in both the target area and the background, right? There's also variation, okay? And so the variation in the background is the background noise. There's also noise in the signal, okay? What I mean by contrast is the difference between here and here, okay? So a lot of people will use the term signal to background ratio, and that has value, but it's important to define what you're describing, right? Or signal to noise ratio. Well, which noise? The noise in the background or the noise in the signal? Because there's noise in each, right? Okay, so that's what that's hopefully explains a little bit what I mean by high contrast here. Okay, let's talk also just a little bit, I hinted at this earlier, in terms of how deep can we go. It's a function of the frequency of the transducer. A higher frequency transducer is going to have shorter wavelengths and therefore less penetration through tissue, right? So in my lab, we routinely work at 40 megahertz. And so at 40 megahertz, I get really, really pretty images. And because I have really, really high spatial resolution, but I also can't go very deep into tissue, okay? Similarly, in the clinic, they tend to work around 5, maybe 7.5 megahertz, okay? So here, the wavelength is longer, or the resolution is worse, okay? So, if we want to improve our contrast problem, so say we want to image between different kinds of mushy goo, so we want to discriminate between liver and kidney here. Well, one way to do that is to use an optical approach, right? Optics tends to have pretty good contrast. So here I have a laser pointer, I'm shining it through a beaker of water, and I have a lot of signal right here, right? And not a lot of signal on either side, okay? So this has a really, really high contrast when I shine this laser pointer through water. However, when I put a few drops of yogurt into this beaker of water, I increase the absorption and scattering coefficient of water, and very quickly, I don't have any contrast. Okay, and add a few more drops of water, and I just have a big diffuse cloud of photons, okay? And you know this from your own experience that it's pretty difficult to get light through your body, right? So what photoacoustics does is it kind of combines the good contrast of optics with the resolution of ultrasound, okay? So this was originally described, the photoacoustic effect was originally described by Alexander Graham Bell. He patented a device called the photophone, and it was going to be an alternative to the telephone and an alternative way of transmitting information, basically by using light rather than electricity. It hasn't been until the last I don't know, 20 years or so that people have been using photoacoustics for in vivo imaging. And that's because the transducers have become more sensitive, the lasers have become faster. So what we're doing here, rather than sound in, sound out, we have light in and sound out, okay? The mechanism is essentially thermal expansion. So normally in most optical techniques, you have some kind of incident, coherent radiation. Unfortunately, when that radiation is scattered, it tends to be no longer usable, right? And one of the great things about photoacoustic imaging is that I don't care how many times this photon has been scattered, because the mechanism of contrast generation is absorbance. Okay, this is my absorber. It absorbs a photon. It has a very short local temperature rise. There's no bulk heating. This is not photothermal therapy. We're not changing the temperature of the animal or the human. This local temperature rise creates a local pressure rise, and these pressure waves back propagate out to the transducers that we have arranged around the sample, and then we can use the information from those pressure waves to reconstruct the image. Okay, so that's pretty remarkable that we can use these kinds of diffuse photons, and from these diffuse photons, we can create a very high resolution image. And the reason is because when these photons, these diffuse scattered photons, create a photoacoustic pressure wave, that photoacoustic pressure wave is not absorbed or scattered, at least it's absorbed and scattered by the tissue thousands of times less than light, right? So if we were trying to do fluorescence where it was light in, light out, right? We would have basically 0 spatial resolution in a system like this, right? You could probably produce some fluorescence, but it would be a big glowing mess, right? Alternatively, when we use photoacoustics, we still have we still have photonic scatter and absorption, but the pressure waves as they come back are basically not scattered at all. So we can pick up a really good spatial resolution as long as we're using a high frequency transducer. Okay, so that's a key point. The other key point is that we can start to do color ultrasound. Okay, so generally ultrasound only has one color channel of information. Even Doppler ultrasound, which looks red and blue, is basically describing the same pressure waves, right? There's no color ultrasound. But what we can do with photoacoustics is excite at 650, 750, and 850 nanometer light. And based on the differential absorbance of the tissue, we can get multiple color channels of information, okay? So the pressure waves that come back will probably all be the same center frequency, but by exciting at different channels, we pick up a lot of information, okay? So that's another really key point. Okay, so I think we talked about this. Generation of pressure waves due to thermal expansion, absorbed light creates heat, periodic light. Okay, so we need a fast pulse. We generally work in nanoseconds, 5 nanoseconds up to about 100 nanoseconds. So why do we need a fast pulse? Well, we talked about the pressure wave is due to this thermal expansion and relaxation, right? So the time of that expansion and relaxation is the frequency of the ultrasound. So to get a higher frequency and therefore a higher spatial resolution, we need to have a pretty fast time of expansion and relaxation, okay? A periodic pulse is going to create a periodic ultrasound wave. The time between the pulse and the ultrasound offers spatial info. Let's see, I kind of want to sketch this out. So if S is your source and this is your sample, so you have photons coming out of here, right? If you have an absorber here that creates pressure waves going back, and you have an absorber here that creates pressure waves, the time of detecting these, so this is both your source and your detector, D for detector, S for source. Sorry, this is my chalkboard. So just imagine I'm trying to show the same thing as I have here. As your light goes down and hits absorber 1, absorber 2, this difference in the time that the light absorbs at these two absorbers is basically negligible, right? The light, because light moves so fast, it's basically hitting these at the same time. However, sound is much slower, right? So this detector can detect the difference between when this sound wave arrives versus this sound wave, okay? And that's how you start to pick up depth information. And that's how you create an image, right? The time between the pulse and the ultrasound arrival offers spatial information, okay? The distance between the object and the transducer is the speed of sound in the ultrasound times the time to travel that distance. Yes, that's what we're showing here. And then again, it's characteristic of the different tissue types. So the time for the sound to pass through the smallest element of interest, which is a voxel. A voxel is just a three-dimensional pixel. The time for the sound to pass through that element of interest is one of the lengths, if you consider your voxel to be a cube, each side of the cube is L. So L over the speed of sound in that medium. Okay, that makes sense. Concurrently, the thermal confinement time, it's the time for the heat that's generated to propagate within that voxel. Okay, so here the thermal propagation time is this same length squared over 6 alpha, where alpha is the thermal diffusivity. Okay, so something like copper is going to have a really high thermal diffusivity. And you can see here what it's based on. Okay. So you have the time for the sound to propagate through your voxel, and you have the time for the heat to propagate through your voxel. And you want your irradiation time to be shorter than both of those, okay? The reason is because it maximizes contrast, because you have good conversion of light to heat, right? If your voxel is already producing a pressure wave, if it's already mechanically vibrating, then you can't expand it again, right? And therefore, you're going to have low contrast if you just have like a CW laser. Well, if you had a CW laser, you're not even going to have the photoacoustic effect. If you had a CW laser, you would start to get bulk heating, right? The reason the reason your tissue doesn't warm up is because this irradiation time is shorter than the thermal propagation time, okay? So if I just took a CW laser and shined it on tissue, yeah, you're going to get some kind of photothermal effect if you've got absorption. And so that's what we're talking about here. You have the spatial confinement. The heat is confined to the area of the absorber. There's no bulk heating. You have time for sound to leave before you're exciting again. So this is acoustic stress confinement, thermal confinement. And so this, which I'm not going to derive in detail, can be used to generate the photoacoustic generation equation.
I have a question.
Sure.
So I was trying to understand, like, so the tissue is expanding and contracting, sort of, or
Yep.
yeah, and contracting. So first the heat has to propagate, and then it will start to like expand and contract. Like, how does it? Is it happening all at once, kind of?
The heat is basically confined to your voxel because the amount of time that you're causing heat is shorter than the amount of time for the heat to propagate.
Mm.
Okay, so you're essentially creating this voxel that's sitting there mechanically vibrating for a very, very short amount of time. Okay, so there's no time for the heat to propagate out to the rest of the tissue because you're only generating heat for a very, very short amount of time.
Okay. And then the expansion contraction is happening as the heat is expanding within the voxel?
That's right. Yeah, so it's the effect that spatially confined heat is causing that mechanical vibration of whatever did the absorbing.
Okay, thank you.
So then we can derive this photoacoustic generation equation that talks about the incident pressure that is generated by a sample is a function of the thermal expansion coefficient. So a material that expands more or less as a function of temperature is going to have more or less photoacoustic signal. It's a function of the speed of sound. It's a function of the specific heat capacity of the absorber. So if you have something that you have to put a lot of heat into in order for it to increase a Kelvin, that's going to have a lower signal, right? It's going to be a function of the absorption coefficient. So if your material absorbs a lot of light, then it's going to be a really good photoacoustic contrast agent. And finally, the incident light fluence. So if you're shining a really powerful laser, you're probably going to get more photoacoustic signal. So people use a variety of hardware designs. So there's both optical resolution and acoustic resolution, photoacoustic microscopy. So here people will take basically a microscope objective and have a very tiny ultrasound transducer in the middle of it. So Li Hong Wang at Caltech is a real pioneer in this space. The systems the system we have at UCSD, we have two systems, use a design like this, where you have a conventional ultrasound transducer, which looks like the transducers I showed earlier, and then you have two light sources on either side. So this light can be a laser, a tunable OPO laser, or it can be LEDs, it can be a pulse laser diode, and those all have advantages and limitations.
Hi, Jesse.
Yeah.
What kind of a laser source you are using for your current setup?
Yeah, there are two. The laser that's in the Moores Cancer Center is an OPO, so it's an Opitec laser tunable from 680 nanometers to 970 nanometers.
It's in the first optical tissue window.
That's right. Yep. Yeah, so there is near IR2. Of course, people do near IR2. We demoed a system for near IR2. The problem is just the water absorption was so high. I wasn't.
Oh, I see. Actually, there is a laser company that they produce a third window, the golden window, from 1600 to 1870 nanometer.
Interesting. Okay.
I'll send you the company's name. Maybe you could have.
That'd be great. We also have LEDs. And so for clinical translation, an OPO, I don't know how many of your students have used an OPO laser, but a tunable laser, right? It's kind of amazing when you think about it, because if you think about the principle of lasing, it should only have one wavelength, right? So a laser where you can say, I want you to change from 680 nanometers to 970 nanometers, that's pretty remarkable. And so, but they do exist, but they're delicate and they break all the time and they're really expensive. And so therefore they're awful for using in a doctor's office. And so the last 2 or 3 years, we've been working on replacing them with LEDs. So the problem with LEDs, of course, is that they have a lot less fluence. So we go from millijoules to microjoules. So, and like I just showed, signal is a function of fluence. And so we have a lot less signal. However, because we are, each imaging event is basically a few nanoseconds, you can have like 1,000 to 4,000 rep rate and average 1,000 frames really quickly and still have it basically to eye look like real-time imaging, okay? So I'm excited about these LEDs because you can drop them on the floor, you can throw them in the back of your car, you can get them hot, you can get them cold, you move them across town, new doctor's office, and they just work. And so, that is kind of the pros and cons of these two different systems. There's also several groups that have designed these kind of array systems where you have light from the bottom and then have multiple ultrasound transducers arrayed around the object. And you can rotate the transducers around the object. So let's see, there's a couple different kinds. We talked about tomography and microscopy. So microscopy, hopefully it stands to reason that you're going to have better spatial resolution, but less imaging depth. as opposed to something like tomography, where you're going to have lower resolution, but better depth of penetration. Okay. And so photoacoustics is kind of cool in that you can use this same mechanism to image from organelles all the way to whole organs, right? So here's imaging melanosomes with photoacoustic microscopy. Here are people imaging individual red blood cells moving through a capillary. Here's a freckle. The scientific term for a freckle is a nevus. And then here's a whole breast imaged with photoacoustic tomography. So you can imagine a breast in here rotating these transducers around it. So that's pretty cool. And then the other thing that's cool here, at least, is we can image the difference between the absorbance of the freckle and the absorbance of the blood based on using different wavelengths, right? So that's how we're getting this color information. Okay, so I guess in the time we have left, I want to talk about just some of the applications of this technology. So if we think about photoacoustic imaging, and so what is actually doing the absorbing? Well, you can have two main bins. You can have endogenous absorbers or exogenous absorbers. So if we think about endogenous absorbers, two of the best ones are hemoglobin and deoxyhemoglobin. And so this is not a very high resolution cartoon, but if you search for the molar absorption spectra of hemoglobin and deoxyhemoglobin, you can find lots of data on that. It's actually a hard measurement to make. I mean, these are still not completely well known, right? And the reason is, as soon as you get hemoglobin and expose it to the atmosphere, it changes, right? So when I was a postdoc, there was this guy who always had balloons full of blood. It was really messy. But people have some general consensus about the absorption coefficient of hemoglobin and deoxyhemoglobin. But what this cartoon does still illustrate is that the differential between the two, and how at lower wavelengths, deoxyhemoglobin absorbs more, and at higher wavelengths, the oxygenated form absorbs more. And then they have an isospastic point around 800. So what you can do is have two, if you excite at two different wavelengths, you have two equations and two unknowns, and you can start to quantitate how much of your hemoglobin is oxygenated in any voxel. Okay, so that's pretty cool, right? But you may say, well, isn't that what a pulse oximeter does? Pulse oximeter is really dumb and easy and has been around for a long time. Well, it's not dumb, it's elegant, but it's been around for a long time. And that's true, but what a pulse oximeter does is it only works in a trans configuration, right? Where with photoacoustics, I can take that transducer and put it on any area and get this map of oxygenation. And I'm creating an image, which a pulse oximeter also does not have. So this is one of the first examples where this is the black and white is the photoacoustic imaging done at around 800, the isospastic point. On the right is the photoacoustic imaging done at around 700, and then also one at 900 and overlaid on top of each other. So here you can start to discriminate between what is the vein and what is the artery, what has oxygenated blood and what has deoxygenated blood. So that's pretty cool. This is an example from my lab where we were using this in the eye. And so the eye is heavily vascularized. The retina has a ton of blood flow. And when that blood flow starts to decrease, or you have some other retinal hypoxia, this can lead to pathophysiology. So, you know, imaging in the eye is very established. You'll probably see optical coherence tomography in this class. There's fundus photography, but none of these are really giving molecular level information. And so this was a project we did with Dan Chow at the Shiley Eye Center here, where we were imaging these rabbits. And so here, the black and white is just the regular ultrasound, and the red is the photoacoustic data laid on top of it.
Jesse, is that because rabbit have very big eyes that you select this model?
Yeah, they have big eyes, and yeah, that's the main reason. And it mimics humans more so than a rat or a mouse. But what we could image is both the muscles around the eye. You can appreciate the lens here by ultrasound, and then you can appreciate this high signal from the retina. But what's pretty cool is that when we go, so this is all at a single wavelength, right? We're just imaging, I think this was probably around 800. But when we go and we do this ratiometric approach, and we put the rabbit on different amounts of oxygen, so 100% oxygen, room air, and 5% oxygen, we could quantitate in the retina how much of the retina is oxygenated, right? There's no other way to do that. So that's pretty remarkable. And then here you can image it in real time. This is a video where we started the animal on, I guess, room air, and then switched it over to 100% nitrogen. So it gets blue over time. So here we're starting, it's oxygenated, and then as it breathes more nitrogen, it turns blue.
Yeah, that's very, very clear. That signal is quantified.
Yeah, and it's mapped, and it's in real time.
I'm curious that if the oxygen level is lower, how is the metabolic activity looks like?
Yeah, I mean, we never kept these animals for more than a couple minutes on lower oxygen. So they responded pretty quickly. But yeah, no, I mean, if they would eventually have died. So, but yeah, I mean, the metabolic activity is definitely slowing.
It's under anesthesia, right?
Anesthesia, yeah, ketamine. Yeah. So, you know, these are gross conditions, right? I mean, these are really dramatic conditions. 100% nitrogen, 90% or excuse me, 100% oxygen, 90% nitrogen. I mean, these are extreme conditions, but it proves the point. And this was kind of, this was the pulse oximetry data. So we could show that the values we determined with imaging at least correlated. The absolute numbers were not the same, but the correlation was strong between the pulse oximetry and the value from imaging. So then we could just repeat this, turn it on, turn it off, turn it on, turn it off, turn it on, turn it off between air and oxygen, and show both the pulse oximeter and the photoacoustic imaging responded.
So is it a survival experiment that after the experiments, the rabbit is directly sacrificed, or you still let it become awake again?
No, these, we could have, it was just too complicated from an IACUC perspective. We never recovered the animals from anesthesia. Yeah. You know, so you might think, well, you know, do we need to worry about shining this laser into the animal's eyes? And this is right at the ANSI limit. You know, people shine lasers into people's eyes all the time for a variety of reasons. And so we were just slightly above the ANSI limit. And so that's another reason why the LEDs, at least for the eye, have a lot of value.
What is the power level of the laser to shine?
This is around 7 millijoules per centimeter squared.
Okay. Oh, this laser is also the OPO laser, or you
OPO laser, yep.
The wavelength is 800?
670 to 980. It's tunable. Yeah. But for these experiments, we were using 2 wavelengths, and I think it was like 750, 850.
What is the pulse width for the laser? Pulse?
Around 5 nanoseconds.
Mm.
Unfortunately, it's like built into this instrument. It's not really a standalone laser.
Oh, it's not replaceable.
No, that's why I bought the warranty because it's had a ton of issues.
5 years warranty.
Yeah, yeah. So to have an example, to talk about something that is kind of a little more clinically relevant, we wanted to look at, could we use this to estimate the intraocular pressure? And so how much pressure is in the eye is actually a very common thing that ophthalmologists measure. And so they use this device called a tonopin, and they basically measure, is your eyeball full of pressure or not a lot of pressure? And if your eyeball is full of pressure, it's occluding those blood vessels in the back of the eye, in the retina. And that makes sense, right? If the eyeball has a ton of pressure, it's hard to get blood into the retina. And so as a model of this, this is an established model. We put a needle into the eyeball and injected PBS. And so we could increase or decrease the pressure in the eye. And so here you can see the needle, you can see the retina. This is a control area that shouldn't be affected. This is, I think, just an adjacent blood vessel. Here's the eye. And so when we start, we have normal oxygenation. So here the rabbit is on room air the entire time. But keep your eye on the retina. And then when we inject, there you can see it, that you can see the injection right here. There, we injected and the pressure goes up and we occlude the amount of blood flow that can enter into the retina. Mm-hmm. And so we could quantitate this compared to the control area and then show that for different intraocular pressure values, well, first of all, this is just proving we can create the model. Different pressures with different amounts of injected saline, and then the pressure correlates to this photoacoustic signal. So higher injection volumes, higher pressure, less oxygenation, and less overall region that even has blood flow. So you can, this is thinner, this is thicker. Okay.
Mm-hmm.
So,
I have a question.
Yeah.
I have a question, like more about the image. Like, has your lab looked into like making the image like have better resolution? Like
Yeah.
how to make it better? Because the retina has so many different components to it, and
Yeah.
I'm just wondering like if you looked into like how to make it, so you can see the different parts of the retina or.
Yeah, so that's one of the things that came up here is when we originally submitted this, we I think in the title we had retina, and then one of the ophthalmologists got it and said, you're actually imaging the choroid, which is an area, an anatomical feature on top of the retina. And so I think we're imaging both the choroid and the retina, and that's why you're seeing these two different areas. Oh, right. Mm-hmm. So honestly, there's only so much we can do. One thing we could do is increase the frequency of the transducer. So this is at, I think, 40 megahertz. So if we went to 70 megahertz, we would have higher spatial resolution. But if we wanted really great resolution, we would use something like this, photoacoustic microscopy. But the problem then is that you just can't go very deep. Okay. So we have
wavelengths, maybe.
Yeah, wavelength. So there's only so much you can do, but those are the knobs, at least that I'm aware of, that people can use to increase your resolution.
So if you wanted to, for example, increase the frequency, then you'd have to figure out a way to make the tissue like so you don't have to penetrate so deeply into the tissue.
Yep.
Okay.
Yeah. Which we generally try to, image what we can
I
rather
see.
than adjusting the tissue. So,
Thank you.
yeah. So, you know, other endogenous materials that other than hemoglobin and deoxyhemoglobin, people have looked at melanin a lot. That's probably the other main one. In the second IR window, people have started to look at fats. That's something that I'm interested in if I get a second IR window laser. But yeah, those are the hemoglobin and deoxyhemoglobin are by far the most common suspects.
So I don't know, I think you already noticed there's a paper published last year talking about the third window, the window from 1600 to 1870. They found that the lipids, the fat, have
Yeah,
more absorption in the third window.
yeah, yeah. Okay, I guess I'm not entirely aware of that paper, but I have seen that these absorbance of fats in those longer wavelengths.
Yeah, if you see the fat, it will be very, very interesting.
Yeah,
Visualize the fat directly.
yeah. Yeah,
And
do send me the name of that company that sells the laser. That would be interesting.
Yes, I will send you an email. So what else, besides the lipids, the fat, and the hemoglobin, what else they would love to look as endogenous signal?
The thing to try to, there's just so much hemoglobin, and hemoglobin is such a strong absorber that it has a way of overwhelming everything else in the body, right?
Right.
And so that's why, you know, my lab and many others have made these kind of exogenous agents that can do molecular imaging through a different mechanism. But yeah, that's the problem, is that the hemoglobin concentration is so high that to try to pick out something else can be more difficult unless it has a really distinct absorption peak.
Mm-hmm. How you avoid for the exogenous, for those probes that you design, how you avoid the signal mixed with the endogenous signal?
Yeah, great question. So it's either you either move your wavelength away from one of the peaks of the absorbance, or it's a concentration effect where you just have a higher concentration, or, you know, a lower concentration but a much stronger absorption coefficient.
Mm-hmm.
Yeah.
So overwhelmingly big absorption or overwhelmingly bigger concentration to get a bigger signal?
Mostly absorption coefficient, because to try to have more material than hemoglobin is hard. Yep. So that's why we like these nanoparticles, right? Because, you know, the molar extinction coefficient of these gold nanorods are like 10 to the 9th. And so they produce a ton of signal at nanomolar concentrations. So plasmonic materials are great for this.
Usually, what kind of shape do you prefer to design for the designing of these kind of particles?
Generally, I mean, we like rods because they're in the IR window, right? That you can have a nice resonance in the optical window, the first optical window. Mm-hmm. You can have shells in the optical window, but those are the most common shapes that we're using if we want to do in vivo work. Yeah.
How big you usually design those nanoparticles?
Let's see if I have this one.
You also did the rats, the bigger in vivo study on rats?
Not rats, but mice and
mice.
So this is an example of nanorods where so gold nanorods have a absorbance in the infrared. You put silver on it and it blue shifts. but you can oxidize the silver selectively, recover the gold, and release silver ions. And so this is regular gold nanorods, and then with a thicker shell of silver on top, you get this color change and you get this absorbance change.
Mm-hmm.
But if you're exciting at 700, you have strong photoacoustic signal, then it goes off, then it comes back on. So what was cool here, we used the silver to treat bacteria, that's neat. But then more importantly is that we could use peroxynitrate and hydrogen peroxide, so these free radicals that are produced by the infection, to cause the silver to oxidize off and recover that photoacoustic signal. So the photoacoustic signal is not just indicating the concentration of these ROS, it's also indicating how much silver has been released. And so that's an example of these kind of plasmonic materials. But these, to answer your question, this is about 40 nanometers by 10 nanometers.
Mm-hmm. Pretty small.
Yeah.
And I think the second particle is more like roundish.
Yeah, exactly. Yeah. So this has the silver shell tends to be more spherical.
Mm.
I should also mention that some of your students may be working on reporter genes if they're like in Peter Wong's group. And so there's been lots of work, of course, on using fluorescent proteins, but a lot of these fluorescent proteins that didn't have very bright signal, because they have a low quantum yield, if you have a low quantum yield, you might have a strong photoacoustic signal. So here's M. cherry that was expressed in the spinal cord of this zebrafish. And so you could, using photoacoustics, they imaged the spinal cord of this zebrafish. And then more recently, people have looked at putting the tyrosinase gene. And so tyrosinase is a protease that allows cells to make melanin from tyrosine. And so it's black, so it absorbs light. And if it absorbs light, it generally produces photoacoustic signal. And so you can see these melanin areas in the cells. And then there's the absorption. No, this is the stability of the melanin. But then, so if you have this as a reporter gene in your cells, you inject the cells and you watch this grow over time. So this is an example of a photoacoustic reporter gene. So generally you want to think of something that is increasing the absorbance. But that's why, to get back to your question, how much do you need? Well, this is pretty low signal. And frankly, here you might have an even better image just with fluorescence using M. cherry.
Mm-hmm. So how they just design it like less fluorescence, just more absorption?
I don't know. I'm bad at these reporter genes. I mean, Peter Wong's lab, I think they still make them, and I think it was, it's kind of luck, at least when Roger Chen's group was making these. I mean, they always wanted them to have a high quantum yield. And at least in the beginning, they were just kind of, I think, arbitrarily changing the amino acids. And sometimes would have a hit.
Yeah, but for photoacoustic, we want low quantum yield.
Low quantum yield, yeah. I actually don't use reporter genes hardly at all in my group. We tend to be more chemical contrast media. I can just briefly mention some of our clinical projects. So this is one where we are building this sensor that monitors heparin. So heparin's an anticoagulant, and when heparin interacts with methylene blue, there's a photoacoustic signal. And so we're putting that on the surface of a catheter, and then the catheter goes in the human subject and you image the catheter ex vivo. And so we're not yet putting that into patients, but we are taking this catheter, dipping it into blood samples from the clinic and correlating that to kind of the gold standard of how fast does blood clot.
This is cool.
Yeah. We've also got stuff we have a trial at University of Southern California in the dental school. So using photoacoustics in the mouth. And then we have a trial at the VA and UCSD Health in terms of using this for wound care. So yeah, I think we're almost out of time, but I just wanted to give my info. One of my best students I got from this class, Yash Mantri. So I don't have the room to take anybody right now, but I'm always on the lookout for sharp master's and PhD students. So if any of you are interested in learning more, there's my contact info. And feel free to reach out. Other questions?
Yeah, any question from students? I think there's a question in the chat box.
More chat to do. Do you know any groups, relation, quantum yield, and photoacoustic response? I think I don't is the short answer. Who would be investigating that?
Maybe from Duke?
Right? Yeah, maybe. It's kind of more, it's a physics question. And so
Leon Wang's previous students.
Maybe, yeah, that would make sense. Or maybe Manojit at NTU in Singapore.
Junji Yao or Song.
Yeah.
Yeah.
Okay, great. Well, again, all of my papers are on the website, and feel free to ping me if other questions come up. But other than that, thanks for organizing.
Thank you very much, Jesse. Yeah.
Okay, great. Well, thanks everybody. Have a good quarter.
Great lecture. And thank you. We can keep in touch. I will send you the laser company's information. And let's also talk more about the research together.
Absolutely. Yeah. I'm on campus a lot, so we can have a social distance lunch.
All right, good.
That's good. Okay, thanks everybody.
Thank you. Bye.